This presentation will walk you through our foundational journey into the world of entomology, the study of insects and other arthropods. Our learning process was designed to be progressive, mirroring the actual workflow of a field entomologist. We began by building a foundational understanding of what we are looking for, then learned how to collect it, and finally mastered the techniques to preserve and identify our findings. To start, Christine will start with the first step in our process, familiarization with our local invertebrates.
In Laboratory Exercise 1, which is External Morphology, familiarization with invertebrates, specifically insects and arachnids, body parts, we examined the external morphology. With that, we identified that they differ in terms of the body regions, since the arachnids have two, which are the cephalothorax and the abdomen, while the insects have three, which are the head, thorax, and abdomen. We also observed the features like antennae, which is present in insects and absent in terms of the arachnids. Also, chelicerae, which is present in the arachnids and vital for the feeding manner. Further, through the aid of the labeled illustrations and e-images, we documented structures such as the compound eyes, segmented legs, spiracles, which is vital for respiration, and jointed appendages. With that, it is essential in terms of the species identification.
After understanding arthropod structures and morphology, we proceeded to study how they are collected and studied in the field.
In Laboratory Exercise 2 titled Arthropods Collecting Tools, we focus on preparing and organizing equipment needed for arthropod collection in both field and laboratory settings. We began by identifying basic materials such as specimen bottles, a fine paintbrush for handling delicate specimens, assorted baits, and proper labeling materials. We also prepared specific collection tools based on habitat and specimen type. For instance, we used a sweep net to capture aerial insects like flies and butterflies, while a bait mixture of tuna and peanut butter was used to attract ground-dwelling insects. Each tool was catalogued and its function clearly documented. A killing jar with ethyl acetate was prepared for safe and human immobilization of specimens, and preservation vials with 70% ethanol were used for a long-term storage. Additionally, we constructed an Iguchi ball, a simple tool used for observing insect behavior, which was particularly useful in this experiment for collecting and studying ants. Overall, this activity emphasized the value of complete preparation and systematic organization, ensuring specimen quality, safety, and efficiency in future entomological fieldwork.
Once equipped with the right tools, we were now ready to apply them in our field collection activity.
In Laboratory Exercise 3 Basic Collection of Arthropods, we applied field sampling techniques to collect and document arthropod specimens. So we used two sampling methods, namely active and passive collection. So for active sampling, we conducted hand-picking and sweep netting around the Gonorrhea's hull, where we collected specimens such as ants, millipedes, and a bee. And for passive sampling, we used a bait mixture of peanut butter and tuna, which effectively attracted ground-dwelling insects, particularly ants, and each collected specimen was carefully positioned and photographed in dorsal, ventral, lateral, and anterior views. So ensuring proper documentation of its external morphology, this process allowed the group to create visual records for accurate identification and future study in medical entomology.
After successfully collecting our specimen, the next step was to prepare them for preservation and microscopic examination.
In Lab Exercise 4, our goal was to understand the hierarchical classification system for arthropods and identify specimens based on external morphological traits. We watched the video, How Do I Identify My Bug Insect Scientist Explains, and reviewed Laboratory Manual 4. Using these resources, we completed a table identifying both provided and captured specimens by order, common name, and distinguishing traits. The provided image of specimen was further arranged into a hierarchical classification table according to the taxonomic ranks. This exercise emphasized accurate identification and the importance of taxonomy in medical entomology. In Lab Exercise 4B, we explored arthropod morphological characters. We reviewed each anatomical term, sourced accurate images, and labeled the corresponding body parts. This allowed us to visually represent each structure and better understand its role in identification. Finally, in lab exercise 4.2, we focused on point mounting small hymenopteran insects. Triangular paper points were made with an entomological puncher keeping the concave side up. Each point was attached to an entomological pin and adjusted using a pinning block. A drop of water-based glue secured the specimen to the thorax between the first and second pair of legs. Using a dissecting microscope, the iguchi ball stabilized the pin while a probe positioned the legs and antennae naturally. Orientation was maintained with the head left, abdomen right, and dorsal side upward, and data and determination labels were pinned beneath the specimen. This produced a cleanly mounted, properly labeled insect ready for microscopic study and preservation. Through these exercises, we gained a deeper appreciation of how careful observation, precise handling, and accurate classification work together in entomology.
This series of laboratory exercises provided us with a strong foundation in entomology by guiding us through a structured learning process. From basic morphology to scientific preservation, we began by identifying the key anatomical structures of insects and arachnids, which helped us recognize their adaptations and roles in different ecosystems. Next, we learned how to organize and use essential collecting tools, highlighting the importance of preparation in fieldwork. We then gained practical experience by collecting arthropods using both active and passive methods around our campus. Finally, we classified, mounted, and preserved our specimens. Overall, these laboratory exercises emphasize patience, precision, and attention to detail, qualities that are crucial in conducting laboratory exercises and future fieldwork experiences associated with medical entomology. This has been our journey thus far in medical entomology. See you in the next one.